meeting is being recorded.
Good evening, everyone. We're just waiting for participants to join, and as they're joining, I can see that. We'll start in just a minute. Okay, well, good evening and welcome everyone to the seminar by Wound Healing Society Education Committee. These are based on the chronicles of wound scene investigation with what was known at that time and where we are now. I will remind you, please utilize the Q&A button to post your questions, which we will be following. We'll take care of the questions at the end, but please feel free to post your questions in the Q&A. So I'll start by introducing myself, and you see the rest of the panel there. So I'm Romana Aslam. I'm the current chair of the Wound Healing Society Education Committee, and very pleased to present this webinar to you. I am faculty at Yale School of Medicine, director of Wound Center, as well as the chief of physical medicine and rehab. And my research interest is in outcome-based wound care. I will pass it on to Kath.
Hi, good evening, everybody. My name is Kath Bogie. I am a biomedical engineer based in Cleveland, working at the Cleveland VA Medical Center and at Case Western. I am co-chair with Romana on the Education Committee and currently on the Board of Directors. Thank you, and Thomas.
You're muted, Thomas.
Hi, I'm Tom Davenport. I'm with Long Island Plastic Surgical Group. I have a practice that is complex wound care and microsurgery. And I do a lot of work with, we have 6 wound care centers that we work with. And my research interest is with skin grafting and biologics.
On to our speaker, main speaker for today, Greg.
Greetings, everyone. I'm Greg Shultz, professor emeritus at the University of Florida and one of the original members of the wound scene investigation team. And we have a lot to talk about today, so I'm going to jump right into the presentation, and hopefully you see my first slide. Is that correct? Everybody agreed? I'm projecting correct? Okay, good. So let me just set the background for you very, very, very quickly here. It turns out that the wound scene investigation team in about 2011 gave a presentation that was basically the chronic wound environment. And so the educational committee thought that as part of the chronicles, this might be a good one to look at and to especially try to understand what have we learned since 2011. So the program that was presented then started out and as typically what we would do is we would present a clinical problem and then the basic scientists would try to explain what we knew and what we didn't know about the problem, and then the clinicians would come in and say how they treated it, and finally we would come back with the basic scientists saying, well, the reason that worked so well is XYZ. So we're going to try to kind of do that same format here, but basically the program in 2011 started out by looking at this chronic wound where there was a epithelium that was not firmly attached and had a lot of instability. And then the clinician, I think it was Lisa Gould at this point, maybe it was Dot Weir, actually did an initial debridement. They used negative pressure wound therapy, and as you can see in the right panel, they began developing really good granulation tissue. The problem was, however, that the epithelium would appear to migrate, but still wasn't adhering to the wound bed very well. And so the solution that was used for this patient was to do an advanced wound dressing with collagen. As you can see in the upper left-hand panel, eventually the wound went on to heal well. So the 2011 program said, why did this chronic wound not heal with conventional therapy? And what we wanted to know then, and what I'm going to talk about now, is what did the field of wound care know and understand about chronic wounds that failed to heal with standard therapy in 2011, and what have we learned since then, now that we're in 2022? Well, very, very quickly for this audience, we have known for decades that we can separate normal wound healing into these 4 phases of healing. The hemostasis, where the fibrin platforms, platelets granulate, they draw in the first wave of inflammatory cells, the neutrophils and M1 macrophages. As I'll show you, they perform the essential role of engulfing and killing contaminating bacteria, but also of secreting proteases that help to remove the damaged matrix so that new collagen molecules can properly insert and convert the provisional fibrin matrix into a scar. Once we move out of the inflammatory phase, then the provisional fibrin matrix gets converted into the initial scar through new capillary formation, through new collagen synthesis. Myofibroblasts contract the matrix. And then finally, eventually, when that initial fibrin matrix is converted enough, then the epithelial cells will proliferate, migrate over it, and we say, okay, the wound's initially healed. But we know that remodeling of that initial irregular scar matrix happens over the next 6 to 9 months to convert the irregular and excessive scar matrix into a more normal dermis, not completely regenerating, as shown in the left-hand 2 boxes. The scar in adults never regenerates the skin, it repairs it, but it's usually adequate. What we've learned since 2011 and had just begun to understand in 2011 was that many of these chronic wounds get stuck in this inflammatory phase. And the key was to understand what was causing that chronic inflammation, what were the downstream problems with that, and especially how to correct it. So one of the keys, of course, is understand that controlled wound inflammation is beneficial. The neutrophils, macrophages engulf the bacteria. They kill them by generating reactive oxygen species, hydrogen peroxide and hypochlorous acid in the endocytic vesicles where they've engulfed the bugs. They also eat these proteases, the matrix metalloproteases and elastase, because those are essential to remove the damaged matrix so that new matrix can be formed properly. Also, we knew that we move out of the inflammatory phase into the repair phase, the granulation tissue, especially in anything other than just an incision, when there's loss of especially the dermis, that we had to form new capillaries, new granulation tissues to replace the dermal tissue and the subcutaneous tissue and the epidermis that was lost. So in the early 2000s and in 2011, the field had put together this concept of the imbalanced molecular environments in healing and chronic wounds. And in the left box, you see that in healing wounds, we had low levels of bacteria, low cytokines, low proteases, reactive oxygen. We had an intact functional matrix that was being produced. The cells were metabolically capable, and there was lots of mitogenic growth factors. What we were beginning to understand was that in chronic wounds, it was the opposite. We had high levels of bacteria that caused high levels of inflammatory cytokines, protease, reactive oxygen. They degraded the matrix and growth factors, and that produced low mitogenic activity in the wound environment. And frankly, the cells tried to proliferate so much they frankly became senescent. What we have learned since 2011 is that we were just beginning to understand, but what we understand is that the bioburden in many of these chronic wounds contains this different phenotype of bacteria. both the planktonic, but especially the biofilm-based bacteria. And so I'll focus quite a bit on that. And again, since 2011, there have been several key papers that have really helped to establish that biofilm components in many of the chronic wounds are a critical factor. And Matt Malone in 2016 reported a meta-analysis that indicated that about 80% of biopsies of chronic wounds contain a classic biofilm structures you see in the scanning electron micrograph of a biofilm on the surface of a wound bed. But as I'll show you, what we've also come to understand that's critical part of biofilm-based wound care is that biofilms are not just on the surface. As shown in panel A, they can also be under the surface of the wound bed. So that has a critical piece of information about how to debride the wound properly. And finally, in panel E, we know that much of the biofilm that forms, up to 20%, can be extracellular bacterial DNA, and that our toll-like receptor 9 actually has evolved the ability to recognize the unique CPG islands that are found only in bacterial DNA to mount an immediate response from our inflammatory cells as soon as a biofilm starts to form. Well, in 2007, this terrific review by DePozo and Patel listed 18 clinical pathologies that they could identify that were really primarily due to the presence of a medical biofilm on structures and in structures. For example, in panel A, chronic sinusitis, our otolaryngology colleagues know that's a biofilm-based disease. In box 13, our colleagues in orthopedic medicine know that prosthetic joint infection is eventually almost always a biofilm. Every catheter or stent that goes through the skin eventually can develop a biofilm and a chronic infection. But note in 2007, chronic skin wounds were not included in this list of 18 clinical bacteria infections that are associated with biofilm. So we weren't quite on the radar screen in 2007, 2011. But as shown in this classic experiment by Carl Schurl in Tom Musto's lab, what was being realized by the film was that the formation of biofilms actually was very able to prevent or delay wound healing. So in the upper right-hand graph, you can see that when they made full thickness punch wounds in the back of healthy mice inoculated with wild-type Staph aureus or Staph epidermidis, they formed biofilms, as shown in panel D, and that there was a significant delay in wound healing. Now, that doesn't prove that the biofilms were causing it. So what their experiment that was so meaningful was they repeated the same experiment, and when they inoculated with the wild-type staph that were capable of forming biofilms, they treated them simultaneously with a quorum inhibiting molecule, the so-called ripeptide, that only prevents the bacteria from converting from a planktonic to a biofilm phenotype. And when they prevented the biofilm from forming, the mouse's robust immune system was able to clear the planktonic infection adequately enough that it did not impair healing. So these data were very fundamental in helping us understand that, yes, biofilm formation can lead to impaired healing, and that it's predominantly due to the chronic inflammation. This was summarized in Bill Costerton's pivotal science paper in 1999, in which he showed that when planktonic bacteria normally contaminate and begin to colonize a wound, our antibodies, our antibiotics, and our neutrophils and macrophages are very capable of controlling that initial bacteria planktonic bioburden, and most of the time will not cause an infection. However, as shown in panel B and C, if the planktonic bacteria attach, begin to quorum sense, convert into the biofilm phenotype, now our antibodies, our immune cells, unfortunately, even our antibiotics are oftentimes very ineffective. And the consequence of that is that inflammation is still accelerated and enhanced, and it's the off-target effects of the phagocytic enzymes, the proteases and reactive oxygen, that are damaging proteins that are essential for healing and lead to an impaired healing. Now, we can measure this again, sort of in the early 2000s, as people began to analyze the wound fluids from acute and chronic wounds, it was noted that in chronic venous leg ulcers, before they got good debridement and biofilm-based wound care, there were high levels of inflammatory cytokines, there were high levels of proteases, and there were low levels of mitogenic activity, that is growth factors in the wound fluid. When those wounds began to convert into a healing wound, moving out of the inflammatory phase because they got good biofilm-based wound care, now the cytokines lowered, the proteases lowered, the mitogenic activity increased, and importantly, the matrix and growth factors, the matrix was preserved, and now the fibronectin, which is a key factor for epithelial migration, survived well enough that the epithelium could begin to move. And also, importantly, the growth factors that were low in chronic wounds now begin to survive because the proteases and reactive oxygen were low. And now the active growth factors begin to replace the degraded growth factors, and wound healing would progress. It turned out that we also began to appreciate a good biomarker for whether a wound is in a capability of healing was looking at the level of MMPs and their inhibitors. And in fact, clinical studies from several different groups showed that the level of protease activities is a tremendous biomarker that predicts when a wound will not heal and when a wound is able to enter into the healing phase. So this really led to this theory of the wound pathophysiology, in which when an acute wound occurs with repeated injury, ischemia, bacteria, mainly at that time planktonic, that they would cause this elevated inflammation, elevated cytokines, they would release the proteases and reactive oxygen, and they would, when they get high enough for long enough, begin to destroy these essential proteins like growth factors and receptors. That resulted in decreased cell proliferation, cell migration, which converted an acute wound into a chronic healing wound. Now, that was kind of the working hypothesis in the early 2000s, and it wasn't wrong. It's just that we didn't appreciate the additional complexity when a biofilm was present along with planktonic bacteria. Now, one of the keys that the field began to appreciate was that when we grow planktonic bacteria like Pseudomonas in suspension culture, rapidly proliferating, mitosing planktonic bacteria, and we treated them with tobramycin, in 4 hours, we could kill every planktonic bacteria there. We took those same sensitive bacteria, grew them in a mature biofilm, and exposed them to that same level of tobramycin for 6 days. The biofilm only lost 1 log. And that was because when the metabolic activity was analyzed in these different mature biofilms, it turned out that in most cases, many of the bacteria are not metabolically active. And antibiotics only kill metabolically active bacteria by interfering with essential bacterial systems like transport, like protein synthesis, like DNA replication. So the key is that biofilm bacteria have a different tolerance to the antibiotics when they're not metabolically active compared to the proliferating bacteria. Well, this stimulated the field to look at what dressings can we use to try to kill these biofilm bacteria if our antibiotics aren't doing very well. So there were in vitro models that were developed that used mature biofilms grown on pigskin dermal explants, and the analysis of multiple kinds of dressings in terms of reducing the level of the mature biofilms after, say, 24 hours of exposure. And in most cases, the antimicrobial containing preservatives in wound dressings would reduce about 1 to 1.5 logs of, say, a 10 to the 6th or 10 to the 7th logs of mature biofilm. Only cadexammer dressing in this model was able to actually completely destroy and kill the biofilm because of the sustained release. Now, the proteases I indicated to you were very important in causing this off-target effect of damage. So again, coming into this concept of how are we going to treat these chronic wounds when we have this elevated bioburden and proteases was the idea of using dressings that would contain collagen. And as you saw in that one previous example, that when the wound was actually treated with a collagen-containing dressing, it began to go ahead and heal well. Well, the point is that the collagen was serving as sort of a sacrificial substrate, and that allowed the growth factors and other essential components to survive. So, for example, if we took PDGF, platelet-derived growth factor, and mix it with chronic wound fluid, after several hours, there was no PDGF left. The protease just rapidly degraded it. If we, however, combined the platelet-derived growth factor and wound fluid with a collagen-containing dressing, now you can see there was only minimal reduction. And of course, something like gauze that doesn't have any substrates for the proteases were not able to really sustain or prevent the off-target degradation of the growth factors, as Lisa Ovington's paper showed. So now there was this movement into using these advanced wound dressings based on what we knew was going to be a problem in many of these chronic wounds, that is the elevated proteases. And in fact, there were good clinical trials that showed that collagen dressings treatment of a wound, once it had been prepared well, would significantly increase the rate of healing compared to standard of care. We also knew, of course, that debridement was important because we weren't killing the biofilms well. And so in the very early data from David Steed's group in the original Regranix study showed that when there was more frequent debridement of the chronic wound beds, both the platelet-derived growth factor as well as just the placebo resulted in a better amount of healing. Again, we didn't really appreciate why that was so important in 1996 and early 2000s, but as we began to understand more about the biofilm formation, we began to understand that that was critical in terms, the debridement was critical in terms of getting the wound into a healing phase. Now, one of the problems is we're making all this big stuff about biofilms. Well, can you see a biofilm on a wound bed? And so in these photos from Matt Malone in Australia, you can see this diabetic foot ulcer over here on the left. Can you see a biofilm on that? Well, no. That's because most biofilms are not visible to the naked eye because they're microscopic structures and a significant amount of them are beneath the wound bed. So on the right-hand panels, you can see that when Matt took a biopsy of that chronic wound bed and actually did scanning electron microscopy, there's no question there was a biofilm component within that wound bed that was probably contributing to the failure to heal. And then Thomas Bjornholt's lab in Copenhagen not only confirmed that there were biofilms, but critically, he showed that the biofilms and the planktonic bacteria could form under the surface of the wound bed. So Staph aureus and Pseudomonas would form anywhere between 30 and 70 microns beneath the surface of chronic wound beds, as revealed by the histology samples using reagents that could detect the biofilm. So this really then helped put together this concept of biofilms. And in this very good review by Steve Percival's group, he emphasized that in a wound bed that was particularly chronic and had stopped healing, that we, one, could see biofilms on the surface of wound bed, but in location 2, we can also see it, as Thomas said, under the surface of the wound bed. Now, the slough, as we'll talk about in a second, is also a potential reservoir for both planktonic and biofilm, as well as the wound dressings, where biofilms can grow if there isn't an antimicrobial preservative present and shed microcolonies back onto the surface of the wound bed. So we begin to understand that biofilms are present in multiple important locations within a wound. Now, more recent technology has evolved where there is a simple wound blotting technique where a nylon membrane with positive charges is just pressed into the wound bed and then stained with a cationic dye that will pick up the negative charges in the wound matrix. And when Dr. Gojiro Nakagami's group at the University of Tokyo did this, they found that if there was any biofilm staining remained, that there was no healing. If there was no biofilm staining, there was an actual improvement in healing in the next week compared to a net decrease in wound area, in other words, or wound healing. So this was confirmed by Dr. Wu's group in Taiwan, where they did, again, this rapid point of care staining, and they found that in cases that had no biofilm staining, 77% of those patients healed in the 1-month follow-up, compared to only 17% that healed when there was still biofilm staining in the wound bed. Now, another technology that has evolved since 2011 has been the use of fluorescence detection of fluorochromes that bacteria make, the pyoverdin and povidone molecules. And when these molecules are illuminated with violet light, they will fluoresce. And this gives clinicians a very good understanding of when there are bacteria present that are missed by clinical signs and symptoms alone, which happens about half the time, and that it would modify the treatment of those wounds when the clinicians knew that there was a significant bioburden problem. And so now what I want to show you in the last minute or so is that there is a new exciting project that's coming on that is focusing on slough. And in 2011, we said, what's this yellow stuff? When we knew to some extent that there were characteristic appearances and context within wound beds when we had slough or fat or fibrin. And we knew indirectly that this wound slough could impair healing because it could help to be a reservoir for bacteria, as we know now, and we're beginning to appreciate, and that it could interfere with the presence of growth factors. So the International Wound Infection Institute has initiated the Slough Project, and Dr. Lindsay Callan and Dr. Matt Malone are the two keys on this. And what this project is doing is photographing the slough. They're taking analysis of the slough and they're analyzing it for bacteria, for biofilms, and especially for proteins, because they want to understand what it is and can it have an impact on healing. And real quickly, I'll just show you initial data from the abstract that was presented at the Wound Healing Society. At the World Union of Wound Healing Societies in Abu Dhabi earlier this month and late last month. And basically, the key here is that when the slough, the loosely adherent yellow slough, was analyzed, it had a high bioburden, polymicrobial. There was no obvious biofilm by scanning electron microscopy. But in the right-hand part, you can see that when they did total protein analysis, they could identify a wide range of proteins. Now, in this patient, it turns out that the red bars are human proteins that are in the slough sample that they collected. And it's interesting that the neutrophil defensin, one of our normal antimicrobial peptides, was most abundant protein in there. And also fibrinogen, which again, of course, we anticipated from understanding the process of wound exudate. But in patient B, now the Pseudomonas identified proteins are in the green bars. And so when they looked at the superficial layer of that patient, they again found a high bioburden. Pseudomonas was cultured. There was no obvious biofilm by SEM, but look now that the protein that was most abundant were from a Pseudomonas gene. And there were other examples of that. And then when they went a much deeper layer, not just superficial slough, but when they actually analyzed slough taken with curette at a deeper level, now the majority of the most abundant half a dozen proteins were all bacterial proteins from Pseudomonas. So this is again beginning to extend our understanding of what slough that it can change based on whether it's superficial or more associated with the tissue. And so in the years to come, within the next year or so, we're going to have a wonderful opportunity to be able to look at more complete analysis of this and really begin to understand more about what the slough is. Now, this is, in my final slide, this has really been integrated into the international consensus guidelines on identification and treatment of biofilms, and we'll hear more about this from Dr. Davenport, but basically we want to start with our most effective treatment to remove the biofilm by debridement, use antimicrobial treatments that will prevent biofilms from reforming. As the wound comes out of the inflammatory phase, we can de-escalate to maintenance debridement. And if the wound begins to heal, fine. Or if we need to add advanced therapies that will stimulate healing, now the wound bed is prepared well, and those advanced therapies will work excellent. So what I'm going to do now is turn over to Kath so that she can give you an update on the pearls for practice that were present in 2011 and in current time.
Thank you, Greg. There seems to be an issue with the slide, at least what I'm seeing.
Yeah, let me just do that. I was gonna.
That looks a whole lot better.
Go.
So no,
Okay.
the current updates were hidden. So as Greg has very thoroughly covered, there's been a lot of work in this area. You know, in 2011, what we knew as practice pearls actually turned out to be mainly correct, but maybe just not quite complete. So we knew that the matrix was essential for epithelial migration, that we needed to get rid of this slough. We're just really starting to figure out why. And we knew we needed to reduce the inflammatory stimulus, and that hyperkeratotic edges needs to be removed. And a lot of this knowledge and the gaps have been filled in as we've got more extensive lab and clinical data. And we've also been able to start to actually build it into something practical, a treatment strategy, as Greg just presented, the step down and then step up. Initiating the most effective treatment first, and then stepping down as the wound begins to heal, or stepping up to advanced wound treatments if it needs extra stimulation to support and heal it. So having just given you a quick overview of the practice pearls and how the research has been able to move it into clinical utility, I'm going to hand it over to Thomas to give us some actual cases and how he implements this kind of approach in his practice.
Great. Thank you very much. So this is an 86-year-old female who had a heel wound from a pressure ulcer. This is a woman who is older, was in the hospital with pneumonia. We've all seen a case like this. And it presents really, and it illustrates a lot or the points that Greg has already made. It was really a truly comprehensive and excellent talk, which really talks about where we are now in wound healing and our approach to the chronic wound. So this wound had been present for months. It had really excellent dressings, and the patient was being taken care of at a facility where she was having regular dressings. And you can see there's no cellulitis. The wound is clean. It is just not healing. And I always say that wounds that are in this situation where they're clean, they're getting dressings, will have a graft of healing that is completely flat for weeks and weeks and weeks. And then all of a sudden, the wound heals. Let me just move to the next slide. But we did not have months and months and months for this patient. It was painful. We have to worry about, it was exposed bone, osteomyelitis. So the first step is really debridement. And this wound had exposed bone, which had infection. It had dead dermis on it. So the first step is removing the dead tissue. Would your body remove this dead tissue over time? And just as Greg alluded to, yes, we have MMPs, we have proteases. Your body is excellent at healing. It takes a very, very long time to remove dead tissue. Those processes are very slow. So what you're seeing here is what would have taken, especially dermis takes a long time, but tendon and bone, if it does happen at all, takes an extremely long time to demarcate. So what would take months, I'm doing in 5 minutes right here. And that is, I'm taking out the dead bone, I'm taking out the dead dermis, and I'm taking this wound and I'm kicking the can down the road and I'm progressing it in just a few minutes, what would take the body many months. So what we see here is we have perfectly clean tissue, has not granulated yet. And then we have here exposed bone. Now this is devascularized. If we did nothing, just like Greg spoke about, a biofilm would form over this area, and we could be stuck in a chronic inflammatory state, and the wound would just stay like that. So what we want to do in this case is we want to get the wound to heal. With this exposed bone, over time, the biofilm would form on the skin, on this exposed wound next to the skin. The biofilm will form on this bone, and the patient will eventually develop osteomyelitis. To avoid that, we added a biologic. What the biologic will do in this case, it can do two things, but in this case, we're using it to just shield the wound, close off the wound, create a neodermis over the bone, which will then allow us to do a skin graft. If there was no exposed bone, the biologic would be to seal off the wound, hopefully prevent a biofilm formation, and then allow the wound to heal. During this time, you would be treating the wound with different topicals, which would inhibit biofilm. So the reasons wounds are stuck, there's two ways. They could be stuck in the inflammatory phase, which we used to think they were just stuck in the inflammatory phase. Well, they could be stuck in the inflammatory phase because there's dead tissue, and they're supposed to be in the inflammatory phase, and that you can fix with debridement. Or they could be stuck in the inflammatory phase because of biofilm, and that can be scraped off and a biofilm treatment placed, and then you'll develop granulation tissue. Once that happens, you could be stuck in the proliferative phase. That means that because of poor circulation, because of diabetes, because of the nature of the area, because of low oxygenation, that wound does not have the sufficient growth factors necessary to heal that wound. And in that case, a biologic must be used. But in this case, the biologic was used to cover the bone. The patient was then skin grafted, and here we see the wound completely healed with just a split thickness skin graft after the biologic was removed. So using all the principles that Greg discussed, we were able to take a wound which really looked very menacing and actually get it to a healed, stable wound just by debridement, control of the biofilm, and in this case, a skin graft. This is another wound that was very, very interesting. So it's a spinal cord patient, paraplegic, chronic ankle wound, and she had a skin graft which failed. But you can see the nature of this wound is very interesting. She's got great circulation. This started off because it was pushing against the side of her wheelchair. She developed this wound, but you can see how it looks like a volcano. It's erupted. This volcanic look is from chronic inflammation. So this is a wound with chronic inflammation, but you can also see there's no dead tissue. It's not like the previous patient. This wound has a chronic biofilm that is preventing this wound from healing. Again, this is, there's no cellulitis. There's, we did an MRI. There's no, at this point, I was sure that she had either a chronic tendon infection or an osteomyelitis, which would also look like this. But this is just a pure inflammation biofilm. And that's why, by the way, the skin graft didn't work. So we performed a skin graft, and that didn't work because putting a skin graft on a stable biofilm, especially a Pseudomonas stable biofilm, the skin graft will fail. So the first thing we did, this is pre-debridement, and we did a debridement. Doing a proper debridement, very, very important. Obviously, what Greg had said is that the biofilm goes deep. And for the first several years of my career, I do not think I went deep enough with my debridement. Why? Because I'm like, geez, I'm making the wound look worse. I'm making things worse. The best thing to do is get rid of all the dead tissue and go deep enough that you are starting from scratch. Now, the biofilm will grow back very quickly, so you also need to use biofilm control treatments as well. So this is the post-debridement. Now, in this case, this wound still failed to progress. So we were doing debridement, we were doing biofilm treatment, and the wound is still failing to progress. What I think had happened here is all of the chronic scar from this wound causes a state of low oxygen tension. And you see this when patients have chronic wounds, it's very scarred, and the scar is not as vascular as a normal tissue, and it creates a low oxygen state. And for this reason, we used a biologic. So there's, again, no cellulitis, no inflammation. We're controlling the biofilm. That still doesn't work. Biologic is placed into the wound. You can see minimal granulation tissue here. Next slide. And this is the wound completely healed. We used all of the principles that Greg talked about, where we debrided all the dead tissue away. Still wasn't optimal. Controlled the biofilm. It was definitely better. But to kind of move it along, we could have done a debridement. And the reason this is actually an area which does have some of that issue with a scar hypoxic state is that there's a tendon and a bone right here, and there's minimal granulation tissue over both. Tendons traditionally have bad blood supply, and so does a bone that you've taken off the periosteum. So this is a low oxygen area, and that's why we use the biologic. Great, thank you.
Great, thank you, Tom. That was great. And showing the practical side of all this wonderful work that Greg was summarizing. So far, I think we've overwhelmed our audience with so much information, but we do have one question, and I believe it's for you, Tom. And the question was from Henry Derby's, was the biologic in the heel wound, did it end up being incorporated into the wound base, or was it fully removed at the end of treatment?
You know, there's many different biologics. Some are collagen based that I use that do not have growth factors. It's basically just the collagen that I use to create a dermal template. This way we have something that would aid in vascularization and create a neodermis for me to put a skin graft on. And the reason I did that is because of that exposed bone. If I try to put a skin graft, for example, I do this all the time on the skull. You have skull, and you try and put a skin graft on the skull, and it's kind of like putting a skin graft on a table. It doesn't work. But if you put a neodermis down first, which actually gets vascularized faster than skin, and you leave it on for a while, it's dead, it's not alive, but it can get revascularization. And then you skin graft on the neodermis, which has a much higher oxygen tension. It's already been revascularized, and that works very well for something that like is skull, bone, tendon. And I use that, and I use many of the collagen biologics for that. For wounds that do not, that have another problem, and that other problem is usually pyoderma, which is a special inflammatory state, or where for some reason the oxygen tension is low, the patient has peripheral vascular disease, the patient has had a chronic wound, the patient's had radiation, then I would use a biologic that contains growth factors. And that could be an amnion or several of the other biologics which have been shown to have a growth factor potential. So basically for me, it's like adding fertilizer to the garden. So the patient lacks that ability and to stimulate stem cells, or lacks the ability to cause a reepithelialization, and that's why I use that kind of a biologic.
Right, thank you. We don't have any more questions at the moment, but I did have one for Greg. You'd mentioned, that biofilm is in every chronic wound, and traditionally 16S sequencing was the method used, which gives you an awful lot of information, but isn't incredibly practical in the clinical setting. And you alluded to the Elysian blue. Do you think that's something that would maybe end up being a clinical practice, or do you think we need something that's even more or should we just assume there's biofilm and go from that?
That's a great question, Kath, and I'll tell you, I think what a wonderful, it depends. I think that one of the key things that needs to become, wherever possible, to be standard of care is to use some of the newer technologies that can identify where planktonic bacteria are with the fluorescence imaging or where the biofilm is with the blotting. And the reason is, when a wound is not healing, one of the key possible contributing factors is the bioburden. And we don't really have a good, rapid point of care way to do that until recently. I mean, in 2011, we certainly didn't have those technologies. So I think the cost of doing the biofilm wound blot is, a couple of dollars. It's nothing. It can be done anywhere because it's a visual read. Any clinician who is taking care of the wound can take the wound dressing off, rinse a little bit, put a membrane on there for a minute and put it in a little tray of stain and destain it and look at it and say, all right, well, there is a biofilm here that we need to get rid of. We need to debride that out. And then you can come back and do a blot again and say, did I get it? Because even the phenomenal clinicians at the University of Tokyo in Gojiro Nakagami's study, half of the time when the clinician had debrided the wound and they did a second wound blot, they found that they had not gotten all of the biofilm. So the key is clinicians need some simple tool to direct them if there's a biofilm present, and if it is, and you attempt to debride it, did you get it? And that's something that can only take a couple of minutes. So it's not a huge, long, expensive, or time-consuming process, but I think it provides a really key piece of information. Literally, that can be done anywhere in the world because you can order the Amersham high bond in plus membrane, and you can order Alcian blue or Ethidium red from Sigma Chemicals. Those are available worldwide. So there's nothing that would be such a barrier that a clinic couldn't begin to incorporate that. Now, the fluorescence imaging is another very important rapid point of care indicator. It, I think, is good because in the study that I mentioned that Tom Serena's group led, in half of the cases when the patient came in and they asked the clinician to do the analysis, and they said, do you detect clinical signs and symptoms of an infection that needs to be removed and dealt with? They said, no. Half of the time they were wrong when they saw the fluorescence imaging. And that information changed what the clinician was going to do for that wound in about half of the cases. So I think the field has moved so much farther than it was in 2011 because we didn't have those point of care tools. Also, finally, then I'll shut up. There are phenomenal new treatments that can be used that laboratory and clinical data are showing are much more effective at removing biofilms. One of the keys that is now being used both in chronic wound care as well as in orthopedics is the negative pressure with installation. And especially if they use installation with a microbicidal solution, not just saline, because that rapidly breaks down the wound slough and it also reduces both the planktonic, but over time will reduce the biofilm burden. So there are some spectacular publications now from orthopedic surgeons, from plastic surgeons, from wound care specialists, where they're using the negative pressure with installation using a microbicidal solution that has made a phenomenal impact on converting that wound from a non-healing inflamed wound with high bioburden into a wound bed that can progress. And as Tom said, then you can put in the advanced dressings, the collagen dressings, the amnion chorion membranes, growth factors, and they work really well because the wound bed is prepared right.
Right. So I think you've answered a question from Naomi Woolhow, which was about what types of commercial products or solutions are best at knocking down biofilms.
Well,
that you're pressure with. Sorry,
yeah,
did you want to add?
I'll cite, because again, we're not trying to endorse any product, but citing published literature, one of the most effective is the hypochlorous acid solutions, not Dakins, because Dakins at pH 9 and 10 has such a high cytotoxicity and a low oxidative reduction potential compared to hypochlorous acid, HOCl, at pH 5 to 6. The clinical studies as well as the lab data shows that the hypochlorous acid has a very low cytotoxicity, and over a period of a couple of days with 8 to 10 cycles of installation and dwell, they just really drive down the bioburden, and they also help to remove a lot of the necrotic tissue and slough.
Okay, thank you. So there's a question from Manuela Martins-Greens. Have you considered that the abundance of ROS and other active species can be contributing to the lack of the ability to get rid of the biofilm? Perhaps decreasing their abundance would also help eliminate biofilm. OS is very important in the bacteria becoming biofilm forming, so the transition from the planktonic to the biofilm. So kind of, how do you head it off at the pass, I suppose?
Well, and again, in the biofilm-based wound care concept, the idea is when you have a good clean wound, don't let the darn planktonic bacteria recolonize and convert into a biofilm. So with Randy Walcott's patients, we published a paper that showed within 3 days of a sharp debridement and no antimicrobial dressing, just wet to dry gauze, the biofilm had fully reformed in all of those patients within 3 days. So the key is get rid of the biofilm as effectively as you can, but don't let it reform. And there are multiple bactericidal barrier dressings that the FDA has cleared. They all will help reduce the potential for planktonic bacteria to recolonize, proliferate, quorum sense, and convert back into a biofilm.
Mm-hmm. Okay, I think we've answered Manuela's question. Get rid of the high OS. And another slightly different question from Daniel Cap. What do you think could be the role of the autonomic nervous system in healing wounds? You know, we've talked about what's in the wound, but what about what's in the rest of the body? Do you have any, either of you?
Well, I'll tell you, you remember about half the time I worked in ophthalmology, so I had a National Eye Institute grant for 24 years where we were studying corneal infections. It turns out that the trigeminal nerve that innervates the cornea is critical in stimulating healing of corneal injuries. And it's due to the fact that the trigeminal nerves synthesize and secrete some great growth factors, not just nerve growth factor, but other factors, growth factors and cytokines that stimulate proliferation of epithelial cells and proliferation of fibroblasts if there's a deep injury. So my guesstimate is that there's something similar also in terms of the sensory nerves providing a enhancement of healing in skin tissues. Now, obviously the cornea has a whole lot higher density of sensory nerves than what, you know, our calf does. But on the other hand, the ability of sensory neurons to synthesize and secrete factors that are critical for healing in the cornea, I think also suggests that they really probably are doing something very similar and beneficial in skin wounds. I think when we look at diabetics that have neuropathies, one of the arguments we've always made, which I think is true, is, oh, well, they can't feel it when they're stepping on their wounds, so they don't get pain. On the other hand, those neurons are not able to secrete the things that the wound probably can use, especially using the analogy of the cornea.
Yeah, that's an interesting concept, and we would be interested. I think it would be valuable to look at it in, as you say, in skin wounds. So, Thomas, did you have anything you wanted to add there from your practical experience or your research?
Sure. You know, early on, I did work with, which is very, very interesting, with Charcot foot, and it's you know, when you see these patients, it looks exactly like a horrible, horrible infection. And it's completely mediated by, or very much mediated by, the autonomic nervous system, and it's a pathological state. So here we have, and the patient then goes on to have a loss of the structure and density and a vascular compromise in just the bony aspect of the foot. And so here we see that. And these patients then subsequently go on to have chronic wounds secondary to this breakdown of the orthopedic structures. And whether there is a component of this type of autonomic dysfunction in pyoderma wounds or other very painful wounds which come on spontaneously, because it's a spontaneous process that has something that people are looking at, and at the university in New Orleans with Dan Lau, we're looking at different aspects of pyoderma and whether there could be an autonomic etiology as well.
That's fascinating. So we're nearly at the top of the hour, and I'm going to hand it over to Romana so she can let everybody know about the books.
Well, thank you. And thank you to all our speakers and the participants. Great discussion, great talk. I just want to say that for the members, recording of this will be available in about a couple of weeks. And then look out for further notice of webinars. They will resume again after the Wound Healing Society and the SAWC meeting next month. I also want to share on the website where you will find the Chronicles of Wound Scene Investigation. I'll share my screen. Let's see here. Can you see my screen or not yet? Hold on. Share, here we go. Okay, so if you go to the Wound Healing Society page, you go under Education and then Chronicles of Wound Scene Investigation right here. And here you have a list of what we have created. You do have the webinar recordings. You have a list of topics that have been changed into flip books. And the video icon is here where you can see the webinar on epithelialization and osteomyelitis. But I'll show you an example. If we have this Charcot foot deformity, it has been made into a flip book, and you just kind of flip the pages like that. And I'll stop sharing. So these are available on the website for all members. And there is a link to the authors if you have any questions. So with that, I thank everybody, and everybody have a great evening. Thank you again to our speakers, and so long, everyone. See you in Phoenix.
Good.
Bye-bye.
Bye, everyone.